start with the, oh no, I would have to start with sharing, right? Let's click on share.
Yep, share first.
And then I guess entire screen, right? Entire screen, desktop 1. Yeah, I think that is it. Yep.
Yep. I see it.
Okay, you see the screen? Great, great, because I got some security error. And now do you see the screen in display mode? So
yes,
in presentation
yes,
mode?
I do.
Oh,
yep.
Good. And then let me see, because I want to then shift to show something in a browser. You see my browser now, right?
Yes, I do.
So this works. You now see the FairDom Hub interface.
Yes, I do.
Great,
Yep.
great. I think we're all set. That was easy enough.
Okay.
So I think there is a bit of a delay, right? I hear there is a bit of a pause in responding. Is that, do you also experience it? It's a bit slow, or is
it's a little bit. It might be my end too. Just before I connected, my local internet service went down for upgrades. So I had to use my phone. Now I'm using my phone as the hotspot.
Okay, okay, yeah.
But it's not too bad.
These things always happen when you really need the connection. Yeah, I'm also I'm presenting from home, so I'm also a bit depending on. Usually the internet connection is quite good, but we share this with a number of other houses, and if everybody starts watching sports on TV, then it might slow down a bit. But I think it seems okay now, so I think we should be okay.
Okay.
Good. Then I
Yeah.
think should I stop sharing that now? And then because
Yeah. Stop sharing, and then I'll put up a one slide introduction thing here in a second.
okay,
Just for people there.
And so because I did not sign up for the seminar, so I just quickly signed in just now. But I think that's okay, right? There is no.
Yep.
Yeah. Okay. Good.
Yep. You're here, so.
And then I'm just gonna. I'll just mute and then I'll see you at 9. Right? Yeah. So you're 11, I think.
Yeah.
Yeah. Good. See you just now.
Yes, that's correct. Okay. See ya. Morning, Herbert.
I had to give a password this time.
I know there's something, I'm not the administrator or something.
And it didn't even work anyway, unless I made it forcibly join.
Yeah.
Okay.
And I couldn't pause the recording or anything.
Okay. Well, Jacky, hi. Can you hear me?
Hi, Herbert. Yes, I can hear you.
Welcome. It's late there, right?
Yeah, it's still okay. It's 9, but I'm glad we
Okay.
could do it. We could do it not at your regular time because
No, that's fine. Yeah, that's fine. Yeah. Anyway, I'm very happy to have you. It was a
Yeah,
talk.
that's nice to do again, to talk again at this seminar series. I think this is the third time now, I think. Yeah.
Really? Wow.
I think I talked first on yeast, and then last year on the yeast oscillations, last year on the Weinberg pathway, and now again something completely new. It's steroid metabolism. Oh.
My memory is going.
That's why you keep all these backups, right?
Yeah, right. I don't have any backups in my head though. Mm. So where are you physically located today? South Africa?
Yeah, I returned on Monday. I was in the Netherlands, but now I'm back in South Africa, enjoying the slightly warmer temperatures here.
Yeah, right.
It was freezing in Europe, and now it's
Well, yeah.
today was 30
Yeah.
Celsius, that is, right? So
Wow. Yeah, that's good.
I don't know what that is in Fahrenheit, but something 90.
No, it's a good. I can do both. I do both these days.
Okay.
Yeah, 30 is good. Yeah, too good maybe.
Yeah, in the sun it's a bit warm. I went for a run in the morning, but that was I was too late. I should have left earlier.
Yeah, right.
But a cat here that is very looking for a lot of attention. So now and then you might see some furry animals walking in front of. Hope he's not gonna walk over the keyboard.
Oh, that'll be good. That'll be all right. So I'll let Bart, you're going to introduce Jacky.
Sure, if you want to. You said you wanted to do it, though.
Oh, did I? Oh,
Yeah, I can.
oh, well, I can introduce him then. Yes. That's fine. So I guess we could start then. It's 1 minute past 11, my time.
Okay.
Yeah. So I introduce Jacky Snoep, a long-time colleague from long ago. I can't remember how long ago now, but he has a joint appointment at Stellenbosch in South Africa, which is a very nice location, and Amsterdam, right?
Right.
In the Netherlands. And of course, those of you who know any modeling or metabolic control will know the Dutch have had a long history in this area. And of course, there's a fairly significant group as well in Stellenbosch too, and Jacky is part of that. And Jacky's been doing, well, he does a bit of theory. He had a very nice theory paper out earlier this year, and also he does an awful lot of modeling. And I consider Jacky to be probably one of the best metabolic modelers in the world. So we're very fortunate. That's right. We're very fortunate to have him speak today. And he says he's spoken to us before, but I can't remember because I'm getting old and I'm losing my brain cells. But yeah, we're very happy to have Jacky back again. He's going to talk on a different topic this time, on steroids. So over to you, Jacky. Thank you.
Thank you, Herbert. And glad you put up the bar a little high to for people expecting now a very nice seminar.
Oh yes, that's expecting an amazing talk.
We have started collaborating or working together a long time ago. I remember I started using Scamp. That was something that you wrote. Oh, yes, you see. That's how
years ago.
it's going on.
Oh wow.
Okay, let me start with sharing my screen and also work in presentation mode. Do you see my screen properly? Okay,
yeah.
thanks.
Yeah.
So maybe now and then if something's wrong, just give me, if you don't hear me or something, just yell. It's about 45 minutes max, right? The presentation. Am I correct? Yeah, I think that was
that's correct.
Okay. So I don't have an awful lot of slides, but there is quite a bit of information on many of them. So we shouldn't be rushing through the slides. That's always nice. And please, you can always interrupt me if something is unclear. If you struggle to follow the seminar because I'm unclear or don't explain something properly, just interrupt me because you might lose the rest of the seminar or if you don't interrupt me. So I'll try and show a modeling workflow that we've been using rather successfully in a couple of studies where we construct mathematical models, usually based on ODEs, and we construct these by characterizing isolated enzymes. And then what we do is we often then reconstitute the pathway by adding the different isolated enzymes together and then follow the dynamics of the metabolites. So we parameterize the model on the isolated kinetics, and then we validate the model by predicting metabolite profiles, usually time dynamics of those profiles. And then, of course, we try and study using often, or analyze these models, often using metabolic control analysis. And then after the analysis, we sometimes in biotechnological applications, we optimize the pathway for a certain function. And in this case, we're going to be applying it to understand some metabolic profiles in steroids in patient serum data. Okay, and so that's quite a long path. So we start with isolated enzymes, then we go to reconstituting a pathway, to tissue, to whole body metabolism of steroids. And we will be focusing on androgen metabolism and then particularly the oxygenated androgens, which is a subclass of steroids that I'll explain that in just a minute, the subclass. So I will have a bit of chemistry, bit of modeling, bit of physics, but the focus is on the model workflow. And before I start, I'll as Herbert explained, I do most of the modeling myself that I'll be presenting. But the experimental work is done in a couple of research groups. So can you see my mouse if I move the mouse? Yeah, okay. So yeah. One of the groups is here at Stellenbosch University, and that's headed by Karl Storbeck, and his group has been active in the enzyme kinetic experiments, and this is usually in iteration with the modeling studies. So what we do is we have an initial characterization, and then I suggest by doing some model simulations to do some optimization of the kinetic parameter estimation just to make sure that all the parameters are identifiable. So Karl Storbeck mostly with on the enzyme kinetics, and also he has been the coordinator of the project. So he has brought me into contact with Wiebke Arup, who is at the Imperial College in London for clinical studies, and that is mostly on the PCOS data. So that's the polycystic ovary syndrome, which I will be touching on briefly at the end of the seminar. And another group is that of Michael O'Reilly, and he is in Dublin, where I'll be, from which group we've received the chronic kidney disease data. Okay, let's take a bit closer look at the system that I'll be talking about. So we look at the adrenal gland that produces some precursors of steroids. That's the 11-hydroxyandrostenedione, but also androstenedione, and the glucocorticoids. So we also get production of cortisol. So what you see here is three classes of networks. We have here in the center is the so-called oxygenated androgens. That will be the metabolism I'll be focusing mostly on. We also have, and they are characterized because they have an oxygen either in a keto or hydroxy form at the C11 of and that's this position. Forget about all this. You can just think of it X, Y, and Z if you're more into modeling. If you're into chemistry, this is the C11 of these steroids. And then we also have the classic androgens, that's shown here with the testosterone as the well-known sex steroid. And they undergo a reduction at the C17 position. So you have a keto group in the androstenedione here, and that's reduced to the hydroxy group in testosterone. And here we have also better known as the glucocorticoids existing in the active form cortisol and then in the inactive oxidized form cortisone. Good, that's a little bit on the chemistry. So basically it's just redox reactions, right? It's either reduction in the C17 going from left to right horizontally, so mostly left to right horizontally, and also reduction on the C11 form if you go up, and then oxidation if you go down. Now the names that you see, the labels here, that are the names of the enzymes catalyzing the reactions. So we have the 11-hydroxy steroid dehydrogenase B1 that catalyzes the reduction of 11-KA4 to 11-hydroxy-A4. But we have then four enzymes here: HSD B1, B2, and HSD 17B2, AKR1C3. Four enzymes, and each of them catalyze multiple reactions, and that leads to some interesting kinetics, right? So what you see, HSD 11B1, it reduces the 11KA4, but also 11KT. And that's just because these things, you might not be able to see the difference very quickly, but the enzymes seem to be mixed up too, right? These steroids are all so similar that many of the enzymes catalyze multiple reactions. And that's all of them catalyze at least 3 and some 4 reactions. And what that makes for interesting kinetics, because if we, for instance, look at the HSD11B1, so if for the reaction, the Ka4 to OHA4, another substrate for the enzyme, 11KT, acts as a competitive inhibitor for this reaction. Because if you look at the reaction, the catalysis of this reaction, and you add 11KT, this reaction will go slower, right? So, and that's competitive inhibition. And what's quite nice is that if we can isolate each of these enzymes and incubate them with a substrate, look at the conversion to product, and then we get the binding constant of HSD11B1 for 11KA4. In a separate experiment, we also look at this conversion of KT to OHT, and then we get a binding constant for 11KT. Now, to now simulate the competitive inhibition of the KT on this reaction, we just add the Km, let's call it the Km value of this reaction for 11KT in the denominator of this reaction. So what we get is we get classic Michaelis-Menten type of kinetics. Almost all reactions are irreversible, but the denominator of each of these reactions is the same for all reactions, and the numerator identifies the specific reaction that it's catalyzing. Okay, that's a lot. I'll show you an example of some of this kinetics later. So what you should remember is we have oxygenated steroid metabolism, classic steroid and androgenic reaction, and the glucocorticoid reactions.
Jacky, just you may mention this again, but do you include product terms?
Yes, so that's what I said, right? In the denominator Okay, yeah. So we have what we test always is, because we will look at progress curves. So we get accumulation of product, and we sometimes start with some product in the reaction to test for product inhibition.
Oh, okay.
So some reactions are indeed product sensitive, but that's only few. So not many of them have product inhibition. Okay, so I'll try and focus a bit on the modeling workflow, and that will comprise of a model construction, model validation, model dissemination, also to show a little bit of reproducibility. How do we actually publish these models? Then model analysis, and then also some applications for these diseases. So let's start with the construction. And so what we saw is that each of these reactions catalyze several reactions. So what we do is we have now a cell line that overexpresses this enzyme, and that is then the only enzyme in that cell line that catalyzes steroid metabolism. In this case, we have the HSD11B2 enzyme expressed, and we look at the conversion of 11-OHT to 11-KT. So what we do is we incubate these enzymes with different concentrations of the substrate. And you see what we do is we vary the initial substrate concentration over 3 orders of magnitude, right? 0.1, 1, and 15 micromolar. And we aim then to vary the substrate concentration from a 0.1 times the Km to 10 times Km. And this is to ensure that we can well identify the Vmax and the Km values for this enzyme. If you do not make such large variation in initial concentration, you will struggle to get good identifiability of Vmax and Km. And an example of that is, for instance, here, what you see is here, if you start with too high substrate concentrations, you get the linear conversion rate. So then you clearly cannot identify 2 parameters on a linear. So you need to vary the initial substrate concentration over several orders of magnitude to be certain that the parameters, in this case, the Vmax and the Km value, are identifiable. And we test this identifiability by making profile likelihood plots to be sure. Okay, now here is
one question, Jacky. You do these experiments in reconstituted system? They're not purified. The enzymes are not purified.
No, but we transfect a cell line that does not express any of the other enzymes. So there is no metabolism of the cell line if we do not transfect that cell line with this substrate.
I see, right. Otherwise, it's basically cytoplasm.
Yeah, but it's correct.
Yeah, okay.
So here is an example of the type of rate equations that we have, and this is again for the HSD11B2, looking at the OHT conversion to KT conversion. So we have OHT as the substrate. So what we typically have is it looks a bit like a complicated Michaelis-Menten kinetics, right? So we have a Vmax times S over Kms divided by 1 plus S over Kms. And then you should, if you ignore these terms, if you put all of these to 0, you have Vmax times S over Ks divided by 1 plus S over Ks. Now these other terms, as I explained before, they are the substrates of the enzymes in the other reactions, right? So the HSD11B2 catalyzes 3 reactions, yeah? And the conversion of OHT to KT, and here you see, here is a product term, Herbert. So this reaction is product sensitive, but it also catalyzes the conversion of OHA4 and Ka to OHA4 to Ka4. So also here is substrate and the product term, and it also uses cortisol as a substrate. So it inactivates the glucocorticoid cortisol to cortisone. So that's why these denominators are so complicated, because all of the substrates of the reactions that the enzyme catalyzes occur in the denominator, because they typically, so all of these are competitive inhibitors with the substrate. Good.
One other question. So I'm going to ask you lots of questions probably.
no,
You said these are oxidation reduction reactions. So those, how does that involve NAD or FAD? Because you don't include that here. So
No, that's correct. So these so the reduction reactions are all use NADPH as an electron donor, and the oxidation reactions use NAD. And this is actually a very good question, because I said that the reactions are irreversible, which is kind of weird because you saw oxidation and reduction reactions. So how can they be irreversible, right? Because then the irreversibility would require a high KEQ. And this irreversibility stems from the availability of and the ratio of the oxidized over reduced form of the electron donor. So what we get, although NADPH and NADP have have the exact same midpoint potential, what you have in these cells is that the NADPH over NADP ratio is much higher than the NADH over NAD ratio. So NAD acts as a good electron acceptor, and NADPH acts as a good electron donor. And I was also a bit surprised on this, but you can simulate these kinetics independently of the cofactor, basically. So the cofactor is always present at the constant ratio.
what about your assays? Do you supplement the assays with NADPH?
Yeah, we make sure that NADPH is in a reduced form by adding a co-expressing glucose 6-phosphate dehydrogenase enzyme.
Oh,
okay. This is getting already quite detailed, but important. And also what you see is that some of these enzymes, if you go to the whole body, are expressed in different tissues. So what we get is the HSD11B1 is overexpressed mostly, is expressed mostly in adipose tissue, and the HSDB2 is expressed in kidney tissue. So I would not be surprised if there is different redox potentials in those tissues as well, with fat tissue being much more in the reduced state, I think. Good. Now, what I've shown here, so this is a typical rate equation which we have for most of the enzymes. And then building the model is actually very simple because we just express all of the ODEs shown here. So we express them as a function of the enzyme activities. So what we get, the change in the Ka4 form is it's converted by HSD11B1, it's produced by B2, and it's also converted by AKR, and it's formed by HSDB2, 17 HSDB2. Good. These are the, so we get then the ODEs, and we have characterized, I show here, only for one substrate, right? One reaction, HSD11B2 conversion of OHT, but we did these experiments for all substrates and all enzymes, right? So that's I say that it's fairly easy to construct a model, but this is 2 years work of just doing enzyme assays, because it's very low concentrations that you have to be able to quantitatively measure. So setting up the assays is quite experimentally. The analytic part is quite hard, but then also doing all the transfections and the conversions is quite a lot of work. And
so I have a question. So can I ask?
Yeah, sure.
So then are all of your reaction rate equations of that same form? So for example, the rate of change for KT, would it then be like a V of KT, but with the substrate being the KT component?
Yeah, so if you look at the different reactions of the B2, you will have that the denominator is the same for all the reactions of the HSDB2, but the numerator is unique. So in this case, it's the conversion of OHT, but there is also a reaction for Ka4, so for F for the cortisol, for the OHA4. So there is a unique numerator for each of the reactions, but the denominator of each of the enzyme, of one enzyme, they all have the same denominator. And indeed, most of the kinetic types are similar to this one. We have a couple that do show some reversibility, but it's usually a very slow reaction in the backwards direction. And then you would have also a minus term in the numerator for that reaction.
Okay.
Yep. Does that answer your question?
Yes, thank you.
Okay, good. All right, so we did the experiments to characterize the enzymes and now have parameterized rate equations that we put in ODEs to run simulations. And that brings us to the model validation. So what is nice with these cell lines that have one of the enzymes transfected is that you can now mix different cells together. Right? So if you have an enzyme that overexpresses the 11-HSDB1, you can also add some cells that have HSDB2 expressed. And this is quite intriguing that these substrates and products seem to diffuse readily through the membrane. Right? So there are no transporters for these steroids, but they just diffuse through the membrane because they are quite lipophilic. So what we did now is we varied the ratio of cells that have HSDB1 or AKR1C3 transfected. So we mixed them at different ratios. So if you look here at the 0, then we have no HSDB1 transfected, but we have cells only have AKR1C3 transfected. And then what we do is we look now at the conversion of cortisol to cortisone as a function of this ratio. And now the symbols are the experimental data with the standard error of the mean, and the lines are the model predictions, right? So this is not a model fit. This is a prediction of the parameterized rate equation for this experiment. And this is not such a strong validation because this is the conversion of cortisone to cortisol, and as you remember, or you might not remember, but in the pathway, this is an isolated reaction here, right? So it's not as complicated here where you have multiple substrates or multiple intermediates. This is a single conversion, but still the model prediction is very good. The other experiment here is where we have the same ratio of these cell lines, but now we look at the conversion of 11Ka4. And this is more complicated because now both enzymes respond on that substrate and the products are also converted. And what we see now is that the conversion of 11Ka4 is predicted quite well. So that's I think that's the red line that's predicted quite well. And also the conversion to the final product, which is, I think in this case, and I should have added some labels, would have been good. I think it's 11-OHT. But what's more is like, how good is the prediction, right? So how good is the yellow line fitting or predicting the yellow symbols? And you see that it's a fairly good prediction. But here, for instance, the green, we do see qualitatively that peak here at a certain ratio, but we don't capture quite the maximum that is reached here. So if you would think, I did not add any confidence intervals in the model, but I think all the model predictions would fall within the standard error of the mean of the experiments. So we did many of these conversions also with adding even more enzymes to reconstitute the whole system. And these predictions look pretty good. We felt confident on a model at that point. Now, after you've constructed and validated the model, it's then important not to just publish your paper and go, but also to make the model available. And I'd like to show you how we do that by putting a bit of advertisement here on the Fairdown Hub, which is a model and data repository or a management platform, actually, where you can also, with research groups, put all your data and models together. And it's very nice that you can keep your data also secure for the group. So you share data with other people in your group, and once it's published, you make it public. It has quite a few advantages. For instance, you can also make the data, you can create on the fly a DOI for your data set. And this is nice because that makes your data also citable. Now, I also always try to actually demo some of this stuff. So let's see if we can actually show this live, how that Fairdown Hub works. So what you do is you type in Fairdown Hub. Okay, let me do that. Fairdown Hub. And it'll bring you to the Fairdown Hub. And then let's just type androgen as a keyword to find the data set, because there's quite a lot of stuff here already in this data management platform. We've been working on this for 15 years, and it's a project funded by Systems Biology networks, and it's driven by Carol Goebel in Manchester and Wolfgang Müller at HITS in Heidelberg, Germany. Okay, the Androgen gave one hit, and if you click on that, you will now get to this study shown here. And what this basically is an overview of the different data sets and models that we've made available for this study. So here is the one on the varying the HSDB1 and AKR1C3 ratio experiments. There are a couple of other experiments, and we'll go through those in the seminar, but I'm not going to show this for everything. But if you click now on one of these files, now you get to an essay, and you can simply download that. This is an Excel spreadsheet with metadata in it. And you can do the same for the model that is published together with this. Download the model, which is in this case a Mathematica notebook. And then once that's downloaded, you can now go to, can you also see this, Herbert? Yeah, okay. Now I simply start that notebook from the download folder, and these notebooks are set up such that it uses the directory in which this file is used. And then you can simply say, evaluate the notebook. So it's gonna read in that Excel spreadsheet, and it makes some data sets from the spreadsheet. I hid the rate equations and the estimation of the parameter values, but that's done on the fly here. And then what you see is if you make some plots, and here you see you reproduce the plots that I've just shown. And these are also the plots that are in the manuscript. So what we do with each of these manuscripts that we publish, we make all the data for the figure available, we make the notebook available, you run the notebook, and then you can reproduce the figures. And for me, that proves reproducibility of the study. And what you'll see is that the parameter values, we also then have a model published at the end. You will see that the parameter values in each of the individual rate equations are still used in the total model, right? So you parameterize the rate equations once, and then you use that throughout the study. Good, that was a bit on Fairdown Hub. Let's see if I can get back.
Question, presumably there's an SPML model somewhere too.
Yeah, so the complete model where we have all the ODEs, we also make that available as SPML, and we also have it on JWS and on biomodels then.
Okay, good.
We typically also produce then ZML scripts to reproduce the simulations of the model, so that you can click on the link in the legend of the figure and it'll load the data from the Verum Hub, it'll load the model from JWS Online, and it'll run the script to reproduce the figure.
Okay.
As you remember, I showed that last year. Good. Then I think that the rest of the talk I'll be focusing on some model application, because I think that's in this case very exciting to see how far can we use these models based on individual rate equations and individual reactions catalyzed by enzymes. How far can we use that to interpret patient data? So we have one step in between here that is going from these mixtures of cells to tissue. So we had access to some adipose cells of type 2 diabetes patients. And then we could incubate that adipose tissue with about 300 micromolar of 11Ka4. And then we waited for 3 days and then measured the different intermediates in the pathway. So what we see is that in the control cell, in the control tissue, most of the 11Ka4 is converted to the 11-hydroxy testosterone. So in this study, we focus on the oxygenated androgens, right? So this is the control cell line. Most of the Ka4 goes to 11-OHT. And what we see is that the model predicts this quite well, right? So the 11-OHT is the major component in the conversion of 11Ka4, and that you should compare that level to this level after 72 hours, which is simulated here. And this is an endpoint 72 hours measurement in the tissue. Now, what we tested here is the effectiveness of a drug that is produced by AstraZeneca. And this drug, it's an inhibitor of 11-HSDB1. And this drug worked very well in rats to bring down the glucocorticoid levels. So HSDB1 activates cortisone to cortisol, the active glucocorticoid. And what was the idea is to, in these type 2 diabetes patients, to bring down the glucocorticoid levels. And this worked very well in rats, but not so good in humans. And that was not quite well understood. And that's why we thought, okay, let's test this same drug now on the fat tissue of type 2 diabetes patients. And what we see, and I didn't even show this, but indeed, the cortisol levels come down drastically if you inhibit HSDB1. So that works. So you bring down the glucocorticoid levels in this tissue. But what we see now here is a large production. Almost all of the 11Ka4 is converted to 11KT. And this 11KT has very strong androgenic activity. So while you bring down the glucocorticoid levels, you activate this androgenic levels. So the positive effect of bringing down the glucocorticoid is negated by the negative effect of producing a highly active androgen. And now the question is, of course, well, how did it work in the rats? And that is then kind of important to know that these. So we have now, if we inhibit this enzyme and this enzyme, we get a lot of this. We do get lower cortisol levels, which is good, but now we get this very active androgen produced here. And rats do not have these oxygenated androgens. So they didn't produce the active androgen. And that's the drug worked, right? As AstraZeneca produced this specific HSD11B1 inhibitor, but it didn't have the desired effects because we got high androgenic activity, and we didn't see that in the rat studies. Okay, that was one application. Another application, and this is now not looking at tissue level now, but at chronic kidney disease patients. So chronic kidney disease is a kidney disease, but it does have strong effect on steroid metabolism. So what we see here is if we measured all these steroids, so and then with a particular focus, of course, on the androgenic steroids, the oxygenated androgenic steroids. We have also the glucocorticoids and also the classic androgens. And what we see here now in this in these charts is that the C is the control group, and then we have 3 other groups, 1, 2, and 3, that have increasing symptoms of the disease. And these have been classified by a marker that is followed in the blood. And this is an estimated filtration rate of the kidney. And that estimation is done by measuring a metabolite in the blood of these patients. So what we see is that some of these steroids change as a function of the severeness of the disease. You see a strong decrease in these oxygenated androgen and an increase here. And don't get mistaken, this is a log axis, right? So this apparently small increase is just as big as the decreases here. And then we also see effects on the glucocorticoids and a little bit on the classic androgens. Now, what we thought of is now, and this is an interesting application of the model. So what we normally do with the ODE models is that we predict metabolite levels, how they change in time, right? You integrate and do numerical integration of your ODEs, and then that will result in the substrate and product changes over time. What we try to do now here is can we estimate on the basis of these steroid levels in serum, can we estimate what the enzyme activities are? And this is kind of inverting the question, right? And normally you would say, well, that's not going to work because you have one steady, and so we assume that these steroid levels are in steady state. So all the ODEs are 0, and you give it certain steroid concentrations, and then can you estimate now the expression levels of these enzymes? And normally, and so you would say immediately, no, you can't do that, because what you know from metabolic control analysis, if you have a steady state for metabolite levels and you get a solution with enzyme activities, if you would increase all enzymes with the same factor, you would get the exact same steady state. So you're not going to be able to get a solution of the enzyme activities on the basis of one steady state estimate. So that's true. But what we can do is we can estimate the relative activities of the different enzymes. So what we do is we see, can we set one enzyme activity to 1 and then get the different expression levels of all the other enzymes? Then you would still say, well, you would not be able to be, I was very skeptical that this was going to work, but it does work. You get a unique solution. Well, you get a best solution. So the expression levels of the different enzymes are identifiable. And that is mostly because these enzymes catalyze the different reactions. So it's not that you just have one reaction going up and one reaction going down. Those same enzymes catalyze 3 reactions with different Vmaxes and Km values. So the combination of the catalyzing several reactions with different kinetic parameters leads to a well-defined best solution of relative enzyme levels on the basis of these steroid levels. Okay, lots of words. Let's look at how we could use that. So what I show now here is what we saw is that only we could set all of the enzymes at the relative expression levels of the control cells. So that's the healthy control cells of non-patients, and the healthy control group. And then vary only HSD B2. and get a good description of the data. So what you have here, each dot here is a person. Here is the control group. And then you see the disease states 1, 2, and 3. So, so what we see is that the model, and that's the black line, can describe the relative levels of these steroids fairly well by assuming that only one enzyme has changed from the control group. Right? So what we see is that this control group has a certain range of HSDB2 activities. Then we have a range of activities in the disease state 1, disease state 2, and disease state 3. Disease state 2 and 3 overlap quite a bit, but there is still a difference. Good, that was quite interesting, right? So it looks as if the only effect on steroid metabolism of chronic kidney disease patients is due to a decrease in HSDB2. This is, if you talk to clinicians, they say, well, that's not such a surprise because HSDB2 is expressed in kidney. So if you now get lower kidney function, it's not such a big surprise that you get lower expression levels of HSDB2. I did start to find out that it's very hard to impress clinicians. But when I show them this plot now, where we now plot now the estimated HSDB2 activities in the different groups, so the control group 1, 2, and 3 disease states, against the marker and the estimated filtration rate in the kidney that is used for estimating that is used as a marker for the severity of chronic kidney disease, we find a very good proportionality between these two. So this marker is not well understood, right? So it's just a marker. If you measure this metabolite in the blood, it gives you an estimate of the kidney function. But this one is now very nice because this is the expression level of HSDB2, one of the steroid metabolizing enzymes in the kidney. So this proportionality did even impress clinicians. And we published this earlier it's last month it came out in the Journal of Clinical Endocrinology and Metabolism. Good. So chronic kidney disease was the effect of chronic kidney disease on steroid metabolism is fairly easy. It's just how stronger the disease, how lower your HSDB2 activity. And you see that in some severe cases, this HSDB2 goes almost completely to 0. And those are they go on dialysis for these patients because they have basically no filtration rate. Good, that was an easy disease. The next one was much harder. This is now, and you see this is a mess, right? So this is now a polycystic ovary syndrome. So each dot is again a patient, and we did the same analysis as before. So I get these steroid levels, and then I try to estimate relative activities of the steroid metabolizing enzymes. And my cat is getting a bit annoyed with me speaking and not paying any attention to him. So what we see now is we had 4 clusters were analyzed by a different group that used machine learning and very fancy principal component analysis that goes way beyond my statistical knowledge. But they could identify 4 clusters, and they were happy with that, and that looked interesting. But of course they had no idea what these clusters represent, right? It's just this machine learning said, here are 4 clusters. So what I try now, can I understand what differentiates those 4 clusters on the basis of the enzyme activities in steroid metabolism? And now what I did is I now could analyze that as a function of AKR1C3. But that's one of the other enzymes in the oxygenated metabolism. So this reduces the C17 form, but of androstenedione onto testosterone, but also the Ka4 to Kt. So so I fitted that enzyme activity, but I also needed now a change in the total levels of androgens. So on the basis of those two, I could separate out fairly good the different clusters. So what we see in this polycystic ovary syndrome patients, some of the patients have a much higher androgenic level and that my model gets that immediately because it starts with the steroid levels as a starting point for the estimation of the enzyme activities. So for instance, this is a control group, and here we have then a group that has a much higher total androgen level. And we have then these two clusters have a much higher AKR1C3 activity. And this is like a 10-fold higher, and this is also, the total levels are also much higher in this group. But it's a subtle difference in total level between these two. Okay, so that was an analysis. Now what can we do with this analysis? And I think that is the advantage of using such a mechanistic model compared to the machine learning model, is that with the machine learning is, here are our clusters, but then now what, right? So, but with the mechanistic model, it says, okay, they have a much higher AKR1C3, and then I could analyze in the graph on the right here. If you have these are the different clusters again, and now with much higher androgenic activities, and I now normalized the different activities to 1 for the different groups. And then I analyzed, although the total activity of the AKR1C3 is different in the different classes, I could simulate now to what extent do I need to inhibit that AKR1C3 to bring the levels down to the control group values. And what we see is that if independent of what cluster you work with, if you bring the activity down to about 20% of the original activity, you go back to the black line is the control group. You go back to the control group levels of this androgenic, the androgen with high activity, the 11-KT form. So I think that's an important lesson from the mechanistic, or an important advantage from mechanistic models, is it gives you a tool how to interfere with the system to bring patient level steroid levels back to the normal levels. Now the graph on the left is was an interesting observation that what independent of what cluster, if you now look at the AKR1C3 activities in these different persons, and we look at the A4 over the T over A4, so that's testosterone over androstenedione ratio, it doesn't vary at all. And this is kind of surprising. And where we see a direct relationship between that activity and the KT over KA4 shown in red, which is what you would expect because the AKR1C3 catalyzes the conversion of the one in the other. It also catalyzes the conversion of the testosterone to androstenedione, but it does that at a low activity. And what we show here is this ratio remains constant because the activity of AKR1C3 is taken over by another enzyme. And that enzyme was not known. So we from this data, we could we could predict that there was another enzyme that has that activity, and we could identify that as actually HSDB1, which is normally catalyzing the reduction on the C11. But what we showed with docking, that it can also do this C17 reduction of androstenedione if it binds in the flipped rotation in the active site. And we subsequently also showed that activity. That's also published last year. That was published last year. Good. So, and it's about time that I finished. Sorry for talking so long with these few slides. Some conclusions that the mechanistic models, even based on individual enzymes, could be used to make interpretations of disease states. And that is, I was very intrigued with that because I was a bit skeptical whether you could actually move over all these hierarchical levels. And yes, certainly you can't do it as simply as this approach, because what we basically assume here is that the whole body is a well-stirred reactor, right? We do as if these enzymes all sit together while they are located in different organs, but it seems to work pretty good. And then those mechanistic models can then indicate points of interference. So what enzyme should you inhibit to what extent to go back to the normal levels of steroids. And we will now start with, we have set up a study to do a clinical study on AKR1C3 inhibition for the polycystic ovary syndrome patients. Now, this is more general, and always separate model construction from model validation, and make your workflow available. Because everybody says, yes, we fit on the construction and we predict, or we validate on the predictions data set. But if you don't make those data sets available, nobody can actually produce it and check whether that is true. Okay, so we understood why that AstraZeneca drug didn't work, and we could correlate the marker that is now used for chronic kidney disease. This is estimated filtration rate, very good, to HSD11B2 levels. The PCOS data was much more complicated, right? You saw that that data set was not as clean as for the chronic kidney disease, which is a much simpler disease in terms of steroid metabolism. And what we needed to not only change the AKR1C3 expression level, but also the total levels of androgens there. Good, that is more than enough. Let me again thank the experimental groups, Karl Storbeck in Stellenbosch, Wiebke Arnold in London, and Michael O'Reilly in Dublin. And this work is funded mostly by the National Research Foundation, which actually pays my salary and my research group. Thank you very much. And I'm open for questions, of course.
Okay, yeah, thank you, Jacky. I have a question, but I'll let anybody else ask first. Any questions from the viewers? No? Yeah, so I got a question. I'm always intrigued by your approach. It seems to work, which is whenever you want to build a model, you first characterize the enzymes, then you drop those characterized enzymes into the model, and then the model just seems to work without any subsequent fitting, which is really interesting. My question is, for the characterization, the other thing, the characterization is interesting as well, because you just do time series. So you spike and then you watch so you don't do a classic Michaelis-Menten characterization where you do all these awful initial rates all the time. How much work is it if you have, so then you do 3 then, yeah. So you only have to do 3 experiments and you pretty much fully characterized the enzyme. Although does this include product effects as well or not? It could do, I guess.
No.
No.
So there is a little bit of a product effect, because if you start, of course, with lower initial substrate, you're going to have a lower final product accumulation. But there is always a strong correlation between substrate and product in these individual experiments. To differentiate and really find out the product binding constant, you need to add at initial condition also some product.
Oh, okay, yeah, I see. So what, can you still get away with just 3 experiments?
Yeah, so it worked in this case. If you have, sometimes you have, we test also for reversibility. So we incubate with the product and see is it converted to substrate, and we then test the conversion of substrate to product at different product concentrations. I just showed it in this case because this. I think the key here is the orders of magnitude difference at initial substrate. So basically, you could see this has 3 initial rate kinetics, right? If we take the slope here and here, that is much more like the traditional Michaelis-Menten.
So if you had this is for a unimolecular reaction. If you had a bimolecular reaction and you wanted to do the characterization, would you do 3 by 3 sets of experiments? Okay.
Yeah.
Okay.
I had a quick question, if I could follow. So when you, I guess, parameterize your model, or when you actually had your final model and you went to the different patient populations, I might have missed something there. I was wondering, did you have to change any rates, or, like if you had a patient that maybe had some type of mutation in that enzyme, would you be able to detect it with your model, or would the same model you think work for that patient population?
True. Yeah, it's a good question. So I doubt whether the model would be, the model is not precise enough to find, say, a small change in the Km value of one of the enzymes for one of its substrates, right? Because what you do is, although you don't change the rate equation in terms of Km and Vmax values, you do introduce a multiplier for an enzyme to estimate its expression level. But that same multiplier is then used for each of the reactions that enzyme catalyzes. So, but that is, of course, that's the way you estimate the expression levels of the enzyme. But that multiplier has, of course, a big effect on the activity. The way that you can still resolve it is that it's the same multiplier for that one enzyme that is used. And what we did with some of the, so for instance, with the tissue incubation, what we did there is, for one of the enzymes, we could use the glucocorticoid conversion, which is isolated from the rest of the steroids, to estimate the relative levels of HSDP1 and HSDP2. And then for the AKR1C3, in that case, we used qPCR, which gave us an indication of the RNA levels at tissue. And then we used that as an estimator for the expression level, which is, of course, also a rough estimate, because RNA is not directly, you cannot directly translate that to expression level. But it worked in this case.
Thank you.
Okay, thanks. Yeah. Any other questions? Or if you let Jacky go to bed.
Yeah.
Okay, thanks very much. That was very interesting. Yeah, I like this general approach, actually. Characterization followed by the model, because it seems to work. It also seems to imply that our biophysical understanding of enzymes is roughly correct, even in cells.
Yeah, so in the cells, I agree. I was a bit surprised that it works so well with different tissues in the body. But we have applied this for a number of different cases, mostly microbial metabolism, so the, say, conversion of glucose.
Yeah.
But also, and that's what I talked about last year, is the Weinberg pathway, which is xylose conversion to alpha-ketoglutarate.
Yeah.
And we had then the purified enzymes and reconstituted them one by one by one. And so you do a sequential enzyme addition, and you get then each of the conversions through the pathway. But we could use that same model to predict conversions in a cell extract. And that was quite interesting, because that indicates that xylose metabolism is quite isolated from the rest of metabolism, because we now simulate that cell extract with thousands of reactions as if it only consisted of those 5 enzymes in the pathway. And that worked very well, also to indicate how to speed up that. And in that case, we used then the reconstitution of a recycling of the cofactors to optimize the pathway, because that's a very it produces a lot of NADH, and then if you again recycle that NADH, you speed up the process strongly, because there was a strong inhibition. So sometimes it works to go from isolated enzymes to cells.
Yeah. In
this case, we went even further to patient data, and that I found very intriguing. So it would be very nice to see if we could, for instance, with PCOS, this is yeah,
okay,
very serious disease for which there are no cures, right? So it would be nice.
Yeah. All right. Well, thanks very much, Jacky, again. I will see you another time.
Thanks.
Okay.
Thanks, Robert. And again, thank you for inviting me. I enjoyed
no
it.
problem. Good. Bye now.
Good.
Yeah.
Bye-bye.
Bye.
All right. I have, for everyone listening, I have one thing I wanted to announce for our next seminar, and it's Professor Anna Niarakis, and she will discuss large-scale Boolean models. So just thought I'd let you guys know, it's this Friday, December 13th. And with that, thank you again for showing up.
I know.
Bye-bye.